kick off. Well, welcome to our Meet the Expert event today, featuring Professor David Thorburn. My name's Tony Cadden and I'm hosting today's session. I'd like to start with an acknowledgement of country and acknowledging the traditional owners of the land that I'm on today, which is the Gadigal of the Eora, and pay my respects to elders past and present, and acknowledge the traditional owners of the lands in which you are all joining from today as well. Just checking to see how we're going. I'd also like to take a moment to acknowledge anyone impacted by mito, either themselves or caring for a loved one. We know how significant this can be, and we welcome everyone today who is from the mito community. My name's Tony Cadden. I actually have a daughter whose picture is on this page, Alana, who was born with mito, and many other mito community members are featured on this page, and many, many more that we support every day. My role at Mito Foundation is as General Manager, and I have the privilege of leading our support services team and to do events like these. And I want to take an opportunity to also introduce Laura, who is in the background today, and many of you will know Laura, who leads our peer support program and many of our events. And if you have any questions today, please submit them via the Q&A function, and Laura will be monitoring those as we go. And the reason why we're doing this topic today is because we know how significant genetic diagnosis is for the mito community. It has been a challenge, and we hear that from a lot of people, that it can be a challenge to get a diagnosis and get clarity on the mito that an individual and their family may have. But we know how significant it can be to receive a label for what is going on for you. And this can help guide your treatment and care. It can provide access to the right and relevant support services across the community, and provide those answers that maybe you've been dealing with for many years. And importantly, it can link into research and clinical trial opportunities. And so we know that diagnosis is not only difficult to get, but there are a lot of people that haven't gone through the process of seeking a diagnosis and may not have a confirmed diagnosis. And this is why we want to talk about this really important project today. Mito MDT has a target to increase diagnosis rates of 70%. And I'd like to welcome our speaker and expert today, Professor David Thorburn. David is a leading researcher specialising in genetics and mito, and his team has helped over 700 people with suspected mito get a diagnosis. David continues to work very closely with the Mito Foundation, which we're very grateful for, generously sharing his time and expertise to support our community, including as a member of our scientific and medical advisory panel and the Mito Foundation board. Thank you so much for being here today, David. We know you've got lots of things to talk about around Mito MDT. Some of the things you'll cover will be in response to questions that we've received from community members. So we'll do our very best to include that throughout. And I think a good place to start before we get into the specifics of the project itself, David, is what's been happening in the world of diagnosis for mito? What do you think has changed, particularly over the last 10 or so years? And what do you think that means for people seeking a diagnosis?
Thanks, Tony. Great pleasure to be here today, and always happy to try to answer people's questions around mito. I've got a few slides at the start to just go through, how the diagnosis of mito and other rare diseases, and also that term rare diseases means conditions that affect fewer than 1 in 2,000 people, and so there's something like 7,000 different rare diseases, and mitochondrial diseases represent about 400 of those. So there's a huge number of different genetic causes of mitochondrial disease. We'll just go over some of the basics of that first, maybe. Is that, yep. So I think you'll all be aware that mitochondria, at least in their, perhaps their primary role, oversimplifying it a bit, but mitochondria are the power plants of the cell, and so they burn fuels, sugars, fats, and proteins with oxygen to generate energy, to transform the food energy into a form of energy the cells can use called ATP, or adenosine triphosphate. And they have lots of moving pieces, if you like. There's over 1,000 proteins in mitochondria that are required to make the mitochondrion work, and a couple of hundred at least are involved in the very central part of energy generation. So maybe if we go on to the next slide, just to talk about the unique thing about mitochondrial disease, or mito, is that in some cases it's associated with maternal inheritance. So it's different to all the regular all the other inherited conditions that are caused by changes in nuclear genes. And a lot of people aren't really aware that mitochondria contain their own set of genes, because the vast majority are in the nucleus of the cell, and there's 23 pairs of chromosomes, about 20,000 genes, and we essentially get half of those genes from mum and half from dad, and so they encode 99.9% of all the proteins in the body. Maybe press onto the next one. And in contrast, mitochondrial DNA is a small chromosome. It is a chromosome, and it has only 37 genes, so 37 versus 20,000, and it's inherited strictly from the mother, and that's largely because the mature human egg has something like 200,000 or more copies of this mitochondrial DNA, and the sperm only has about 50 copies, and some of them get in, but there's a nice destruct system to stop those paternal mitochondria being transmitted for maybe what are theoretical reasons, but so mitochondrial DNA is strictly maternally inherited and present in multiple copies. So there's lots of things that can go wrong to cause mito. And on this slide, I'm not sure if, oh no, I haven't got my pointer's not going to work, is it? But down in the bottom right part of that slide, you'll see some things called complex 1, or C1, C2, C3, C4, C5. And that's the core part of the energy generating machinery that you might have heard of as the electron transport chain, or the respiratory chain, or oxidative phosphorylation. And those are the primary, that's the primary issue that we think about when we think about mitochondrial disease. But there's maybe 200 proteins required for those complexes to work, but there's a whole lot of other things going on in the mitochondria that we need to work. And so that includes maintaining the mitochondrial DNA, replicating and expressing it, the whole protein synthesis system inside the mitochondria that's different to the protein synthesis in the rest of the cell. And we have to import a lot of proteins, add in cofactors. So that's why we pretty rapidly have got up to more than 400 different genes that can underlie mitochondrial disease now. And so I don't want to dwell on the detail, just to say that there are all sorts of things that can impact, all sorts of inherited problems, if you like, that can impact on mitochondrial energy generation. So if you go on to the next slide, and that's just a cutout from a review we wrote 6 years ago now, listing some of the different genes that had been linked to mitochondrial disease. The ones in red are some of the ones in the mitochondrial DNA. And you can see there's all sorts of different categories. The ones directly involved in oxidative phosphorylation, or OXPHOS, and ones that impact secondarily. So there's hundreds of these genes that are required for energy generation to work properly. And so mitochondrial disease are thus far more complicated than, say, something like cystic fibrosis or the hemoglobin disorders, where basically many times the symptoms that prompt suspicion of those conditions are very clear, and there's a single gene that can be tested. It's been much harder with mitochondrial diseases because one condition, for example, Lee syndrome, or Lee disease, that some of you will know about, has quite specific brain changes and a sort of clinical pattern. But there's about 100 different genes in which changes in the DNA, what we call variants, or what used to be called mutations, can all cause that same clinical presentation of Lee syndrome. So having a clinical diagnosis of Lee syndrome leaves open a huge bag of genes to look at. And so what I'll talk about in the next couple of slides is this amazing revolution in genomics that has happened over the last, I guess, 15 years, and its impact on how we go about diagnosis. So maybe the next slide, Tony. So I'll explain a little bit more about what genomics is in the next slide, but basically it's about, instead of looking at one gene at a time, just looking at all our 20,000 odd genes at once. So, you know, certainly prior to 15 years ago, and even still, you know, an individual suspected of mito, you know, most of the time the diagnosis used to rely on something like doing a muscle biopsy and hopefully finding something, and then gene by gene studies where we targeted one gene at a time to look at. And just next one, Tony. So basically what happened was, you know, clinical assessment and some widely available laboratory studies, perhaps prompting a neurologist or a geneticist or some other physician, cardiologist, to think about mitochondrial disease, and that might prompt genetic testing. But up until relatively recently, you know, there were a very limited number of things that were widely available. Testing for some of the common, well-known mitochondrial DNA mutations, for example, in blood or urine, or some very specific presentations maybe going to a particular nuclear gene. But in general, it would involve, in adults and kids, a muscle biopsy, and then that was looked at by histology, sort of cutting through the fibers and staining them for mitochondrial function, or measuring those OXPHOS or respiratory chain enzymes I talked about. And then if you got a hint from that it was really mitochondrial disease, it might guide sequencing of specific genes to try to find a variant that was underlying disease. And it's really hard to put solid numbers on this sort of concept of, you know, how many patients can you diagnose, because you don't really know how many patients there are. But, you know, I guess we'd say that prior to 2010, 2015, we were maybe only able to get a genetic diagnosis or a genomic diagnosis, find the right gene in 10 to 20% of patients. And so what's happened since about 2010 is that these new technologies came in as part of the Human Genome Project that was published in 2003. So these technologies allow us to, you know, initially, we did a study back in 2008 with colleagues at the Broad Institute in Boston, when if you were thinking about trying to sequence a whole genome, it would have cost about $1 billion or something, probably. It was just completely unrealistic. And the first study we did with this sort of technology was taking samples from 100 patients, pooling them together and sequencing 100 genes, and then trying to work out which patient changes were from which individual. And so then, you know, costs kept coming down, and we got to what's called exome sequencing, where we sequence the known genes or panels of genes, and then costs kept coming down, and now the logical thing, particularly for mitochondrial disease, is genome sequencing, where we're sequencing all the nuclear genes and the mitochondrial DNA. And many of you will know that there's now an MBS item number, Carolyn Sue led the case for that to be funded, and that's a great thing, because now it means many individuals can access genome sequencing. And I guess what most people would say is that many of the studies in mitochondrial diseases and other complicated groups of rare diseases, we're probably getting about 50% of the diagnoses now following genome testing. So maybe just from a blood sample, getting that diagnostic yield of 50%. But unfortunately, a lot of patients end up either with no findings that are thought to be relevant, or with what we call variants of uncertain significance. I'll go into those a little bit later, but basically there are changes in the DNA where the level of evidence isn't enough to say this is definitely a cause of disease. So I'll talk a little bit more about the technology, and I don't want to go too deep into the science, but yeah, people sometimes talk about the central dogma of biology or of molecular biology, which is that DNA, if you like, is the blueprint. It makes RNA, which is the message to get translated into proteins, which in general are the things that do all the work. And so this slide, and I'm sorry if it's a little bit blurry, I've borrowed it from a review by someone else, and it's showing the double helix of DNA at the top, and studying DNA is using this technique called genomics, where we're sequencing all of the 3 billion base pairs of DNA in a human or sequencing other animals. And basically, this review sort of suggested that DNA tells you what can happen. It's, we use a whole range of analyses, and if a particular change in the DNA variant's been linked to disease before, we might be confident that that's causing disease, but it's not. It's telling us it's a change, and we have to use other information to see if it's relevant. So genomics was the first omic technology to come in, and is now available in most countries and in most states in Australia, for example. And the DNA encodes RNA, that's ribonucleic acid, and instead of being 20,000 genes all in one place, the RNAs are individual messages for each gene, and they're called transcripts. And so the omic that we use to measure them is transcriptomics, and not all RNAs are expressed in every cell. This kind of level is what makes a heart cell different to a brain cell, and a blood cell different again. And so basically with RNA, we're not necessarily testing all 20,000 genes. We're looking at the RNA that is detectable in the sample we analyze, which might be blood, for example. And so looking at the RNA tells us a bit more about what appears to be happening rather than just what can happen. And then the next layer is the RNA encodes proteins, which are strings of amino acids that fold up into these complex patterns. And we can use a technique called proteomics to measure those. And because these are the things that are doing most of the work in a cell, the proteomics, we can argue, is saying what makes it happen. So with proteomics in blood or cultured skin cells, we might be able to detect, say, 8,000 different proteins and a very large number of mitochondrial proteins, about 800 or so, and see if they're present in the expected amount or not. And then the next one, so again, this particular review was about another technique called metabolomics. And so proteins such as enzymes, that are involved in making ATP work on these metabolites. So molecules like ATP and lactate are metabolites, and metabolomics is the omics that measures those. And again, this is a little bit biased by the people who've reported it, but they were interested in what has happened and is happening by measuring the metabolites. And so that flow of information and generating the working components of the cells together can cause a phenotype or a clinical presentation. So that's just sort of a general background, if you like, to what's happened over the last 15 years. For most of my career, my lab, the clinical part of my lab has done these enzyme assays in muscle biopsies or in cultured skin cells, and that has guided us to genes we were familiar with or novel disease genes, but we were doing it one gene at a time. Genomics has transformed things so that we can now get a diagnosis by looking at all the genes at once in maybe half of all people. And for about 10 years or so, I've been trying to say that I'm wanting to put my diagnostic lab out of business, because who wants to do a muscle biopsy if you can get a diagnosis from a mil or so of blood?
Thanks, David. That's a lot has certainly happened in the last little while, and in the last 15 years in particular. And it's really exciting to see how much easier it is for people to get a diagnosis that is really accurate. Noting, as you've said, and you'll talk about, there's still some situations where that's not happening yet, but the potential is huge. And so how does this, all this background, which is really impressive and really interesting, quite complex, but very interesting, how does that relate to your MitoMDT project? And can you tell us, what is it all about?
Yep.
And what do you think people really want to know about it?
Yeah, so once again, I've got a few slides here. I don't have slides for every question, but just to explain it visually, that probably helps. So if we go on to the next one. So MITO-T is a national project that was funded by a grant from the Medical Research Future Fund, and it involves expertise across Australia and international collaborators. So the goal of this project was basically part of a call from the Medical Research Future Fund to ask to lift the diagnostic rate or the diagnostic yield in individuals with rare diseases to over 70%. And so our project has been recruiting patients with suspected MITO and applying these sort of innovative interdisciplinary approaches to try to get to an answer. So on the next couple of slides, I'll show that it involves clinical collaborators across Australia to recruit adult and paediatric individuals, and incorporates the expertise of 6 very experienced Australian labs to apply these genomic and other omic tests. And so there's a range of things we do. They include looking at the clinical genomic sequencing data, looking at it in a research context, and I can explain the difference there later on, but it's basically opening up the box to look at more things that may not be clinically proven, but might be the answer. And then going on to these omic technologies or targeted tests. Sorry, if you go back one, sorry, yeah. So that just listed the clinical network that was proposed in the grant at least, and not all of these individuals have ended up being active in the project. I don't think people realize the challenge of running these sort of multi-site projects and getting governance at individual sites and getting everyone to agree, including the hospital, the university, the research groups, is really quite challenging. And so we proposed a clinical network that should be able to access patients from around the country, including Tasmania and the Northern Territory, as some of the state genetic services interact very closely with those. And so I won't read through all of those individuals. Sean Murray's listed there with the MITO Foundation. He's obviously not a clinician, but Sean was a chief investigator on this grant and has been very involved with it on the way through. So on the next one, yep. So this lists the main research and diagnostic sites, and just going around from the top. We're very involved with an organisation called the Centre for Population Genomics, which is based, it's really split between the Garvan Institute in Sydney and the Murdoch Children's in Melbourne, led by Daniel MacArthur, who's one of the best known genomicists in the world, and Kaz Simons, who's got a really pretty stellar track record in rare disease research. Then at the Colling Institute, University of Sydney, Ryan Davis, who's very involved in studying biomarkers and genomics. In Melbourne at MCRI, John Christodoulou and I lead a lab, and Alison Compton is our head of the genomics team, and our former PhD student, Nicole Lake, who's now running a group at Yale University, have been very involved, particularly in the genomics and in some functional studies. At the Bio21 Institute of University of Melbourne, there's 2 groups, David Stroud and Daniela Hocker, forming one focused on proteomics, Di Stoyanovsky, another one focused on metabolomics. At Monash University, Mike Ryan and Luke Formosa, again, some of the best known mitochondrial biologists in the world, and done a lot of work in the proteomics area and other methods. And then Alex Filipovska and Stefan Sira in Perth, again, internationally renowned, particularly in the area of transcriptomics, but with all the omics really. So there's varying expertise in those sites so that we could get true international quality expertise for all these different technologies.
Thanks, David. It's amazing to see so many experts involved in this project. So tell us about how people were involved in the project and how they recruited, what was the eligibility for them, and how many people were actually involved.
Yeah, so the eligibility for this project, again, in a research project, you have to define pretty strict inclusion and exclusion criteria. And we relied largely on criteria called modified Nijmegen criteria, which split they basically generate a score of up to 12, with 5 to 7 being probable mitochondrial disease and 8 to 12 being definite mitochondrial disease, if you like. But this is intended for use before a gene has been found. So it's really the level of suspicion. And that score has up to 4 points for sort of clinical features, up to 4 points for imaging and metabolic features, and up to 4 points for other biochemical and genetic testing type features. So that basically, eligibility was defined as a score of 5 or more for individuals who didn't have a confirmed genomic diagnosis that explained their clinical features. And for individuals who didn't meet that score of 5, but the team agreed that there was a strong clinical suspicion of mitochondrial disease, we also included a number of those patients. So we were having basically initially monthly and then fortnightly multidisciplinary team meetings reviewing 4 or 5 patients at a time. And this slide was from April. We're up to that time, we had 104 individuals recruited. Unfortunately, recruitment has now stopped at the end of May because the grant finishes in August and we have to finish off analyses. And so we finished with, I think it's 125 individuals. So we had a good run in the last month or so. And again, partly because of these reasons of governance and obtaining governance and having an enthusiastic clinical team who could refer the patients, fill out the clinical information in what's called a REDCap database and so on. It ended up that Queensland has had the majority of patients referred from the Queensland Metabolic Service, or the Lifetime Metabolic Service there, so children and adults. In New South Wales, we're only able to recruit from the Sydney Children's Hospital Network, in Victoria from Royal Children's Hospital, and the Victorian Clinical Genetics Services, which has links into most Victorian hospitals, as well as Northern Territory and Tasmania. South Australia, the Women's and Children's Hospital, and in Perth, the Perth Children's Hospital. So that was showing 104 patients. The final number is 125. But yeah, we've had to stop recruitment at the end of May, basically because the patients, once they're accepted, then need formal consent into the project. They do pre-consent to allow to have their de-identified information considered by the committee, and then formal consent and obtaining samples, often from, if it's a child, the child and the parents, for genomic and other studies.
Thank you. And so given the distribution here and the main sources, were most of the participants children?
Yeah, in the end, unfortunately, we were hoping to have about 50-50, but again, there's a number of reasons contributing there, but the paediatric referrals were, if you like, able to be organised earlier, and very enthusiastic teams doing that. And so we've had relatively few adults. It's in the order of 10 to 20% of the total.
Yeah. So you've come to the end of recruitment, obviously still a little bit left in terms of the actual project, but what can you tell us about the highlights and successes of the project so far?
Yeah, so in one sense, it's still early days, because the genomics on well, only about half of the patients has been done. And then the proteomics has been done on quite a few of those patients who are unsolved. The transcriptomics, the RNA studies, are still largely being waited on. So we haven't, it's not like we have shown we can diagnose 70% of the patients as yet, but of the patients who've been studied, there's about 10 or a dozen so far where the proteomics has allowed us to gain a diagnosis that we couldn't get from just the genomic testing alone. And just because it's a little bit more advanced in our hands, I guess, the proteomics has been the big highlight, including being able to do what we call ultra rapid proteomics, where traditionally, with genomic testing is amazing, but typically in clinical setting, it's often taking maybe 2 to 3 months to get an answer back. In genomics, teams around Australia and internationally have refined that down so that it's possible to do it in 3 days, particularly for kids in intensive care unit and so on, and generate a result. But if that's, and that has a profound impact, lots of cost savings in terms of management and unnecessary investigations, and obviously ending those diagnostic odysseys rapidly. But again, maybe half of those are unsolved. And with the proteomics, we've also been able to get that down to a 3-day timeline when needed, and be able to solve kids who are still in ICU, or do rapid turnaround for example, in 2 cases. One case where a woman was 6 weeks pregnant, and another where family had lost a child at something like 20 weeks of pregnancy, and genomic testing suggested a likely diagnosis. And when that was sent to us, we could turn it around within less than a week to be able to inform the patient, have those variants of uncertain significance upgraded to causative, if you like, and then that opens up the reproductive options for the family. So I guess just in the last couple of weeks, it was quite exciting that we had a paper published, with bookended there, if you like, by Daniela Hock and David Stroud and myself, as sort of the lead authors, describing this untargeted proteomics enabling ultra-rapid variant prioritization in mitochondrial and other rare diseases. And this is really showing that the proteomics method is better than what I've been doing for 30 years in the lab, doing this respiratory chain enzyme testing, and suitable for implementation into the clinic. And that's what we're trying to do now. But that's quite a process, so it'll be a couple of years before I think we will be successful with that. And one of the great things about proteomics is it works really well for mitochondrial disease, but about half the known rare diseases have proteins that are expressed in samples like blood or skin fibroblasts, cultured skin cells. And so it's effectively a generic method for a very wide range of conditions. Whereas in the past, depending on what you found in mitochondrial disease, there might be 20 different tests that could be relevant, and you'd choose the most relevant one. And maybe there's 2 labs in the world that offer it, and it depends on when there's a PhD student who can do it, and so on and so on. So we're really excited about this, because it is allowing us to push up that proportion of individuals who have not achieved a diagnosis following genome testing, but with proteomics that can be done. The transcriptomics or RNA sequencing does the same thing to some degree as well, and it's a bit more advanced in clinical terms, but most of inherited diseases, the changes don't actually affect the RNA in a way you can measure them. So proteomics, we think, has very high potential in the clinic. And you can see that study involved a lot of collaboration across many sites. And I think on the next one, we got a bit of news a few weeks ago in late May, with something like 85 news outlets covering it, and the Guardian in the UK, with comments from UK researchers saying this needs to get into the National Health Service, and a commentary published with the paper from someone from the Children's Hospital of Philadelphia, making the argument that this, again, needed to get into clinical testing. So certainly from my perspective, the proteomics has really advanced as part of this study, and part of other studies as well. So that's probably the biggest highlight, that it's allowed us to give diagnoses to families, in some cases quite rapidly, and in many cases just providing the final answer there.
Yeah, that's really significant, David. So congratulations to you and your team for all the work, and we're excited to see what continues to come out of the project. You mentioned that maybe it might take a while to get into clinical practice. What do you think the potential is there?
I'm confident we will get it into clinical practice, but it's quite challenging. And it took quite a number of years to get genomics moving from being a research test to a clinically validated test, and then longer again to get funding for it, things like MBS rebates and so on. But and that's partly because traditional testing in labs is often only measuring one thing at a time, and when you're measuring thousands or millions of things at a time, it's much harder to prove one change is significant. And so that's to a degree the challenge. But we've been able to take advantage of some great collaborations. There's a program at the Royal Children's Hospital in Melbourne called Happy Kids, I can never remember the acronym, but it's something about harmonizing pathology testing. And so that program has two nurses who coincidentally sit outside my office, and they have this program where in the past, when children were tested for biochemical or other tests, often the reference ranges that were used were just guesses from adult individuals, because it's been a bit more ethically challenging to get samples from enough children to show that what is a healthy range, if you like. The Happy Kids project approaches people who are in hospital, often for a minor surgery or other things, and there's an amazing degree of altruism around people at places like Children's Hospital or the Royal Women's Hospital in Melbourne. And so very often those kids might have a line in, and they give permission for a mil of blood to be taken. And so we were able to access blood from 400 children with no suspicion of having an inherited disease, 10 boys and 10 girls from 0 to 1 month of age, 1 month to 12 months, and then every year, which is just an amazing resource. And those samples are currently being run on the mass spectrometers as a huge batch to enable us to generate much more robust reference ranges. So that is a key to being able to offer the proteomics testing. We're also we have things in Train where we should be able to access substantial numbers of adult patients as well. Again, in a children's hospital, my focus has mainly been on pediatric disease, but these techniques work, very well for many of the adult onset presentations as well. So, we are seeking to implement that test into the Victorian Clinical Genetics Services, and collaborating with centres in the UK and US for them to implement it as well. And David Stroud's team has gone to a lot of effort to have a user-friendly website where labs can look up whether their protein of interest is expressed in blood cells or skin cells or wherever, and look at our control data, which at the moment is only 40 blood samples, but will expand to 400, and compare to some of our results on individuals with a problem in a particular gene and so on. So we are very positive, and have a lot of support from people that this is a bit of a missing link in increasing genomic diagnostic rates.
Thank you. I'm sure I'm not alone when I say we look forward to that. We look forward to just anything that improves the diagnostic rate, and they improve the number of conditions that are identified. So we had a few questions come through that were about genetic testing and understanding genetic testing. And you've kindly put together a bit of information around that covers some of those questions, including trying to understand a bit more about variants of uncertain significance. Can you talk us through those?
Yeah, so I've got some information on the next slide, I think, which there was a question that was sent in asking about the Medicare item numbers for whole genome sequencing. And I thought I'd maybe just go through that and explain the diagnostic process a bit, because one of the things with genomics has been that it's really increased the guidelines and approaches that people use internationally now for diagnosing inherited diseases, because once you bring in this kind of technology, there's a lot of potential for finding things that might not be relevant. For example, in a child in ICU, it's not relevant at that time if they're a carrier of a BRCA2 breast cancer mutation. It might be relevant to the family and so on. Probably not while the child's in the ICU, and just guidelines around incidental findings and approaches. So in clinical labs around the world now, genome sequencing, exome sequencing, or mitochondrial DNA sequencing are analysed with rules around interpretation according to guidelines from the American College of Medical Genetics and Genomics and the American Molecular Pathology Group. And they have gone through a number of iterations to improve things and get insight from other countries implementing them and so on. And so, to a large degree, clinical labs study the data they get in pretty much the same way. And when we do genome sequencing, it's basically lots of reads of letters or bases in the DNA. And for nuclear genes, we essentially read each position at least about 30 times or more. So that gives enough confidence that when we see, say 15 reading the common variant and 15 reading a different one, we can be confident that one's come from one of the parents and one from the other. So it has sufficient data for quality metrics. And the mitochondrial DNA is read at much higher depth. So we're typically reading each of the mitochondrial DNA bases about 2,000-fold on blood. So that means for people who are familiar with the concept of heteroplasmy, or the amount of the mitochondrial DNA that has a disease-relevant variant versus the healthy variant, we can estimate that heteroplasmy in the sample very accurately from genome sequencing. Exome sequencing, not you can find mitochondrial DNA variants in, but it's not nearly as robust, and so genome sequence is ideal for mitochondrial DNA and nuclear gene discovery. But there's so much information in that sequencing that you have to think about the level of effort that labs go to. And obviously this is highly automated now, and there's lots of algorithmic support for it. But typically, when the clinical question is, does this patient have mitochondrial disease, we're not looking at all 20,000 genes. A clinical lab will focus, in the first instance at least, on the approximately 400 genes that have been reviewed and regarded as having a clear link to mito. And so those are informed in Australia by a website called Panel Australia, where it was led by Zanetza Stark from Victorian Clinical Genetics Services, but has reviewers from around the country, and is now, I think, in place in all the genomics labs in Australia. There's great collaboration between diagnostic and research labs in Australia. I think we probably have the best integrated system just about anywhere. The UK is very good as well and better funded, but basically everyone uses the same gene lists, and so those gene lists have been informed by trained individuals looking at the evidence to decide, is this enough people with a similar clinical presentation to be confident that the changes are caused by changes in this gene. And it's informed by expert panels from an organisation called ClinGen. So, for example, John Christodoulou and I sit on a couple of ClinGen expert panels, and usually at about 6 a.m. or 7 a.m. We meet up once a month for each of the panels with colleagues in the US or wherever, and go through the evidence base that for particular genes or particular variants. So those kind of endeavors, if you like, focus in on what genes you're going to look at. For mitochondrial disease, it turns out that probably about a third of the time when patients are referred clinically, although their symptoms might sound like mitochondrial disease, roughly a third of the time it's actually due to a change in another gene. It's not mitochondrial disease, it's a change in a gene that has an overlapping range of symptoms, or an overlapping phenotype, what you might call mitomimics. So many labs, and again, I'm familiar with VCGS, or most familiar with VCGS, because it's on the same floor as me in the Murdoch Children's. We'll also look at relevant gene lists. So when patients are referred, their key features are provided in a documented way in what's called the human phenotype ontology, or HPO terms, which allows for those descriptions being unambiguous and staying with the sample throughout its journey. So those other gene lists might be looked at. And very recently, VCGS and some other labs have started to introduce an automated reanalysis program, which to me is amazing, that every month now unsolved patients' data are reanalyzed. And I think there's been 200 to 300 patients that have been solved by that, because we just keep on generating, we keep on generating better data around, here's a novel disease gene, or here's evidence on this change, and that can now be automatically incorporated. So maybe press the next one. When we talk about individual changes in the DNA, when we do whole genome sequencing, there's probably going to be 1 million changes in any individual from what you might call the reference sequence. And so we have to look at them to try to decide which ones are relevant. Daniel MacArthur, now in Sydney, developed this database called Nomad, which now has data from over 100,000 individuals who've had whole genome sequencing. So you can look at variants and decide, well, if they're present in 20% of the population, they're probably not going to be a disease-causing variant that is, penetrant in causing disease. So we look at the population frequency. There are various bioinformatic predictions of likely severity, and when the diagnostic labs report something, they also are expected to download it into an international database called ClinVar. So there's a lot of sources for looking at variants, but nonetheless, a lot of the time what we find are variants of uncertain significance. And so to be called pathogenic, which is clearly disease causing, effectively we need to have 95% certainty that that's correct. To be called likely pathogenic, it needs to be 90%. To be called benign or not relevant to disease, 5% certainty of that, or likely benign. And so we've got this huge range of variants in the middle that are called variants of uncertain significance. And if those are to be used in clinical decision making, there's a very strong incentive to look for evidence, such as using omics, to upgrade those variants from uncertain significance to pathogenic or likely pathogenic. There was a question in one of the community things also about, will diagnostic labs release raw data of results to patients? And typically what diagnostic labs do is generate a report focusing on the key changes. If a clinician seeks that raw data, then that can usually be provided. But I'm not aware of any labs in Australia that would provide raw data to an individual from clinical testing, because the professional ethos is that it needs interpretation by an expert team.
Yeah, thank you. And we certainly recommend that individuals ask questions if they're not sure about their results, and go back to their referring doctor, but also the value of talking to a genetic counsellor as part of that process. But what I'm hearing here is there's lots of different ways to increase the rates of diagnosis. Even when maybe a while ago there was no result, there might be an opportunity to get a clearer result using some of these new technologies.
Yes, the technology, and also communication's just been improved too, I guess, so much. And as an example, we have done testing on patients with suspected mitochondrial disease for a number of years in various cohorts, including some retrospective analyses. I'm perfectly open about this. Some people might feel it sounds spooky, where we've had a waiver of consent to, if we've received a sample for diagnostic testing, we don't just put it in the freezer and forget about it. We consider whether it can go on to further testing, and with approval from the ethics committee have done that. And last year, I should say the results from those sort of studies are put into a confidential database called run by the Centre for Population Genomics, where the individuals are anonymized and so on. And last year we had a query from that team, as we get periodically, about a change in a gene, which a group in the UK had found a change in a few patients and thought it looked suspicious. And nowadays you can phone a friend, or you can put things in a website called Gene Matcher and get put in touch with groups who are also interested in that same gene from studying an individual with suspected features. And very quickly that went from a handful of patients to 140 patients written up in one story with a completely novel mechanism of disease, one of whom had been thought originally to have mitochondrial disease. Actually, a few of them had a suspicion of that from other sites. Again, just this overlapping phenotype thing. And, in the past, you just didn't get that sort of organized way to contact potentially anyone who's found an individual with a change that looks interesting, as well as the phone a friend still works really well for knowing people who can do things quickly and that you can trust.
Yeah, that sounds really promising in terms of people. We hear it quite often of going through the process and not getting a result, particularly people that have maybe done it quite a while ago. So yeah, recommend that if people are interested in seeking a genetic diagnosis, there are pathways. Yeah, reach out to your specialist, or reach out to our helpline, and we'll point you in the right direction for your local service. And it's really exciting to see what's possible. You've got another slide that was helping to answer some questions as well, and we'll throw it open to any questions that are coming through. I'll give you a bit more information there, David, to explain.
I just put that on in case people ask. So quite often people ask about children versus adults, and there are a difference in the causes. So and this is an old slide again, so it's only saying 300 mitochondrial disease genes. Say 400 is a better estimate now. So what we and others have found in substantial studies, and we published a paper on this just at the start of this year on a previous study called the Mito Flagship, that when you look at children with suspected mitochondrial disease, about a quarter of the time it's due to changes in the mitochondrial DNA, more than half the time to what are called autosomal recessive conditions, where mum and dad are each carriers for a condition but are healthy, and the child has the 1 in 4 chance of getting the copies that don't work so well. And then small numbers with X-linked conditions, which typically affect males more than females, or autosomal dominant conditions where you only need one change. We're probably still underestimating the autosomal dominant ones because of some of these things about them not all being penetrant, where you can get carriers who are not affected. So anyway, in children, 25%, maybe up to 40% in Lee syndrome, but mitochondrial DNA mutations are the minor cause rather than the major cause. Whereas in adults, and again, there's maybe some bias in these numbers, but traditionally we've said that probably 2/3 of the time disease is caused by mitochondrial DNA mutations, with some of those other mechanisms contributing.
Thank you. So just looking at some of the other questions that came through, and we'll take a couple of minutes for questions and then we'll wrap up. So when somebody gets a genetic test, we've got a question about, and this is for a child, but I think it relates to children and adult, is how does someone know how much of an individual's mitochondria are affected based on their genetic testing?
Yep. So I'll assume that's asking about mitochondrial DNA, because on genetic test report, every lab that reports on nuclear gene changes would say whether it is what's called homozygous, so both copies have the same variant, or if there are 2 copies, and if they know if one's from mum and one's from dad, and so on. So with nuclear genes, it's very clear. For mitochondrial DNA, at the moment, VCGS have been offering whole genome sequencing and being able to pull out mitochondrial DNA sequencing. I know a number of other labs have been working towards it, and I'm not sure if anyone has achieved that yet. But say on a VCGS report for a mitochondrial DNA variant, it would say in the report how much of the mitochondrial DNA variant is present, whether it's 10% or 20%. And there might be some caveats around that, because for some of these, particularly in adults, what you find in blood may be much lower than what's present in muscle. I saw there was a question about MELAS before, and that's really the classic one, where the common MELAS mutation disappears from blood from birth at 1 or 2% a year. So by 30 years of age, it may have gone from 70% down to say 20 or 30 or 40%. It's possible to actually correct for that. Back in the old days, where our testing wasn't as sensitive, those would potentially be missed if it was, say, less than 10%. Genomic sequencing is a much more sensitive test. And so different diagnostic labs use different levels, but basically VCGS, I think, reports variants down to 2 or 3% level. And if it's what's regarded as a pathogenic variant, if it's less than that, they'd still comment on it. So with genomic sequencing, we're much less likely to miss those kind of mitochondrial DNA variants that go down. But the genome sequencing, all the mitochondrial DNA sequencing that we've been doing for 10 years or so using these technologies, but just looking at mitochondrial DNA, are really exquisitely sensitive for measuring the level of heteroplasmy.
Excellent. I don't think we've had any other questions come through. So I'm going to throw to a question about whether you're aware of any research for the PMPCB gene.
Yeah, I think in these scenarios it's better not to go into specific detail that can be identifying, but just as you know, for any of these genes, and there's PMPCB is one of the less common nuclear gene disorders, and so there's maybe a handful to 20 patients or something that have been reported internationally, and so it's probably been less studied than others. But basically, for any gene, there are likely to be ongoing research studies somewhere on these novel disease genes or less common ones, because people want to sort out exactly what they're doing. That particular one is known to be involved in processing of proteins that have been imported into the mitochondria, and so there's a bit of knowledge about it. And there's expertise in that process in Australia and elsewhere for functional studies.
Thank you. All right, we're coming to the end of our time. I'm not aware of any other questions coming through, but please, Laura, let me know if that's not the case. I want to thank.
Sorry. Looks like there's one just came up, I think, okay, on how to get a clinic diagnosis when tests don't show that symptoms linked to mito. Do you want me to answer that very quickly?
Yes, please. Thank you.
So, there's not one answer, but as I mentioned, when we investigate patients who are thought to have mitochondrial disease, maybe a third of the time it turns out to be not mitochondrial disease, but something that looks like mitochondrial disease, very similar overlap. So, again, I guess a lot of people might be confident they think they have mito, or their clinician might be confident they think they have mito, but until they get a genomic diagnosis, it's not proven.
Yes. So is why we're really supportive of projects like MitoMDT and others, as we're advancing diagnosis at a genetic level, and that is so important to really understand the condition and to drive research. So anyone who's interested, or anyone at any point in their diagnosis journey, you've got any questions about that or how to go about it, jump on our website. We've got some resources. There'll be some more information on that in a moment, and you can always call us on a helpline. But I just want to thank David, Professor David Thorburn, today for your really comprehensive discussion around diagnosis and your MitoMDT project, which is really, certainly for me, and knowing that there's been lots of other techniques needed to get a diagnosis that are far more invasive and far more time consuming, this is really positive, and I think should hopefully be really positive for people that are still looking for a diagnosis. So thank you very much for your time today.
Thanks, Tony. Pleasure.
I want to thank everybody for joining us today. We do have some resources related to genetics and mito on our website, and we also have information about clinical studies, which is something we're really focused on connecting our community to, because that's a way to learn more about different types of mito, but also hopefully fast track access to therapeutics and all sorts of different kinds of treatments that are coming in the pipeline as we learn more and more about mito and how to support it. Sydney. So thanks everyone for joining today. Really appreciate your time. Thank you very much, David. This information will be available at some point on our website if you want to go back to it. But if you've got any questions, please reach out. And I hope everyone has a great day.